Today we'll start with our chapter number 2, which is biomolecules. So we have already discussed biomolecules in the previous session, but one thing that I was forgot that basically in your biomolecules, this chapter of biological molecules are called biomolecules. In your AS level, a lot of knowledge is required. In that PDF which I have downloaded, it does not consist of enough information you requires at the level of AS. So at AS level for biology 9700, which is the code, you are required to have a lot of knowledge of your biochemistry or biomolecules. So the content we are looking for is water, carbohydrates, lipids and proteins. This is the very foremost essential chapter for you candidates because of only reason that if your this chapter is very weak, it means that you are not able to score a steady grade. Okay. So now what is the thing is that you must be able to determine the structures, understand about some bonds, and we'll be looking more about this as knowledge. Okay. So first of all, okay, at least 80% of the mass of living organisms is water. Balanced diet, whenever you see in your balanced diet, uh, these of the elements are arranged accordingly to their proportions. And almost all the chemical reactions of life take place in aqueous solution. The other chemicals that make up living things are mostly organic macromolecules belonging to the fourth groups: protein, nucleic acid, carbohydrates, or lipids. Okay, and then these macromolecules are made up of monomers as shown in the table below. Between them, these four groups make up 93% of the dry mass of living organisms, the remaining 7% comprising the small organic molecules like vitamin and inorganic ions. So we will look for the group name is protein, nucleic acid, and carbohydrates. The monomer of protein is amino acids, so we'll be discussing either it's amino acids or amino acids, whatever. And for nucleic acid, the monomer is nucleotide. Okay, what is a monomer? That is the question. We'll be discussing it. Okay, and for carbohydrate, the monomer is monosaccharides, and these polymers names are being written here with the dry mass percentage. Okay, so the dry mass in the human body of protein is 50%. It means that the foremost important thing in the in the balanced diet is protein, and then nucleic acid is 18%, carbohydrate is at 15% dry mass in the human body. Okay, and the lipid is 10%. Okay, the components of lipids we are discussing here is that fatty acids and glycerol, and the largest units are triglyceride. Okay, so here we are with the water. We will be discussing water, and water molecules are charged with the oxygen atom being slightly negative and the hydrogen atom being slightly positive. As you all know, what are these? Okay, these opposite charges attract each other, forming hydrogen bonds. Now, this is called the hydrogen bond. Okay, hydrogen bonds are present where uh, these are weak long distance bonds that are very common and very important in biology. Yes, hydrogen bonds are the first bond which occurs in the reaction after which the covalent or either ionic bond react. Okay, so before covalent and ionic bond to be formed, there must be a hydrogen bond to be formed. And how does it form? It is shown in the diagram below that there are the two oxygen, not oxygen molecules, these are the two water molecules where the oxygen is partially negative and hydrogen is partially positive. So now what you can see here is that the oxygen, which is a partially negative, it will be attracting towards the hydrogen, which is a, and this line, which is the dotted line, we show the hydrogen bonding by the dotted line. Remember this in your mind, you can write it as a note, and after which you can also the covalent bond is also visible here. Remember that the covalent bond is shown here by the straight line with no gap, by the dotted line showing the hydrogen bond. So whenever the question or the examiner asks in the question to show the hydrogen bond, you must use a dotted line to show it. Okay, and this is how one molecule of water is bonded hydrogen to another molecule of water, and through the hydrogen bonding, the each of the water molecules are bonded to each other to make the water, basically. Okay, water has a number of important properties essential for life. Many of the properties below are due to the hydrogen bonds in water. Okay, we'll be discussing here the solvent, specific heat capacity, latent heat of evaporation, density, cohesion, ionization, pH. All right, so we'll be just discussing it fast as we are required to move ahead. Solvent, okay. Because it is charged, water is a very good solvent. Understand? Because it is charged. Charge or polar molecules such as salts, sugar, and amino acids dissolve readily in water and so are called hydrophilic. Hydrophilic means water loving, where the word hydrophobic means water hating. They are non-polar or they are uncharged. Uncharged means there is no uh, partially positive or negative sign on them. As you can see here, there are many of compounds which are non-polar. All right, means they are, for suppose we can take the example of chlorine molecule. Chlorine molecule is polar or non-polar. When two chlorine atoms combine to make the covalent bond, there must be partially positive. No, both of the chlorine atoms are partially negative. It means that there is no opposite charge attracting to each other. It means that they are non-polar. Alright, you can take a more of example, and many of molecules are also non-polar, which are uncharged. Alright, that's why they are hydrophobic. Hydrophilic and hydrophobic are the word which are very important for you in your AS level and your A2 level. This will be discussing for you candidates in your which we have already discussed, yes, in your chapter number 4 or chapter number 3, which was the cell surface membrane. Yeah, it was chapter number 4, your cell surface membrane, where we have discussed what is the meaning of hydrophilic and hydrophobic. All right. Okay, specific heat capacity. Water has a specific heat capacity of 4.2 joules. Okay, so this is the SI unit, which means that it takes 4.2 joules of energy to heat 1 gram of water by 1 degree Celsius. This is unusually high, and it means that water does not change temperature very easily. This minimizes fluctuations in temperature inside cells, and it also means that sea temperature is remarkably constant. Okay, latent heat of evaporation. Water requires a lot of energy to change state from a liquid into a gas, and this is made use of as a cooling mechanism in animals, sweating and panting, and plants transpiration, which we'll be discussing also. Not in this video, but there is a chapter for you guys, and I hope that I'll be just making a live stream soon on that chapter also. As water evaporates, it extracts heat from around it, cooling the organism. Density. Water is unique in that the solid state ice is less dense than the liquid state, so ice floats on water. This will be also discussing in our AS chemistry. Yes, we have a good concept for it in your states of matter in your AS chemistry. As the air temperature cools, bodies of water freeze from the surface, forming a layer of ice with liquid water underneath. This allows aquatic ecosystems to exist even in sub-zero temperatures. Alright, cohesion is also in your next upcoming chapters. Water molecules stick together due to their hydrogen bonds. So water has a high cohesion. This explains why long column of water can be sucked up tall trees by transpiration without breaking. It also explains surface tension, which allows small animals to walk on water. Ionization, we will be discussing it in AS chemistry. When many salts dissolve in water, they ionize into discrete positive and negative ions, such as the sodium chloride, where the sodium is partially positive, plus the chlorine, which is the partially negative. Many important biological molecules are weak acids, which also ionize in solutions such as acetic acid, acetate. All right. The names of the acid and ionized forms, acetic acid and acetate, in this example, are often used loosely are interchangeably, which can cause confusion. You will come across many examples of two names referring to the same substance, such as phosphoric acid and phosphate, lactic acid and lactate, citric acid and citrate, uh, pyruvic acid and pyruvate, aspartic acid and aspartate, okay, etc. The ionized form is found in living cells. What is pH? The power of hydrogen. Water itself is partially ionized, as you can see from this equation. All right, that hydrogen is partially positive and hydroxyl group is partially negative. So it is a source of protons where the hydrogen positive ions or positive ions. And indeed, many biochemical reactions are sensitive to pH. Okay, you are not required to learn this log. Pure water cannot buffer changes in hydrogen ion concentration, so is not a buffer and can easily be any pH. But a cytoplasm and tissue fluids of living organisms are usually well buffered at about neutral pH, means 7 to 8 pH. Okay, just let me see what else is remaining. Okay, so we are moving towards the carbohydrates. Let me see one thing. Okay, we are moving towards the carbohydrates, and in the carbohydrates we are required to see the monosaccharides, disaccharides, polysaccharides. All right, the term macromolecules. Okay, carbohydrates contain only the elements carbon, hydrogen, and oxygen. This is also understandable by the name as carbohydrates, where the hydrates means OH. Okay, hydrates means like water. Yeah, the thing which consists of a water molecule is a hydrate or hydrated, where the carbo means carbon is present and hydrate means that hydrogen and oxygen both are present. Okay. One more trick which I'll be gonna telling you later in this video is that for your carbohydrates formula, the includes monomer, dimers, and polymers as shown in the diagram. That carbohydrates. Okay, carbohydrates are sugars and polysaccharides, while the sugars can be divided into two sections, monomers and dimers, where the monomers of sugar is monosaccharide and dimer is disaccharide, where the word mono means one, di means two, and saccharide is the sugar. Alright, now the example for the monosaccharide is glucose, fructose, and ribose. It means that the okay, what is the journal formula for the carbohydrate is the bracket open CH2O bracket close and then the n which is the number of carbon. Okay, so this from the ribose, fructose, and glucose, you can see that the number of glucose is C6H12O6. It means that the number of carbon, if the number of carbon is 6, then the number of oxygen is 6, and the number of hydrogen will be double of 6. Okay, so 6 multiplied by 2, and then you'll be getting the answer 12. We'll be discussing glucose, fructose structures in this chapter. In the disaccharides, sucrose, maltose, and lactose. Okay, we are required to know about them. And in the polysaccharide, the starch, cellulose, and glycogen. These three terminologies are also most important. We'll be looking at it because most of your paper of paper 1, paper 2, and your paper 4 sometimes is completely organized by the biomolecules, and there's a lot of structures shown to you and ask from you the identifier. Moving towards the monosaccharides, and here's the general formula. Okay, now you can see that this is the glucose, or it is a monosaccharide glucose, and okay, which is the now just see one thing is written here very important. Forms a six-sided rings. The as you can see from your what is your right? Okay, it means that it is your left, maybe left or right, where no carbon and hydrogen is shown to you, only just hydroxyl group or one. Okay, I will just required to zoom in. Okay. This is shown to you and you're required to just focus on this, that in the ring structure you can see that oxygen is only at the side of ring. Other than that, no of the side of the ring is mentioned. It means this is carbon. This ring or the corner of the ring is showing you the carbon. The zigzag, this is showing you the carbon. Okay, so carbon number 1, after oxygen molecule, the carbon number 1 is having the hydroxyl group below it, okay? And then the carbon number 2, carbon number 3, and again then carbon number 4, you can see the hydroxyl group below. And then carbon number 5, okay, is shown to you. And carbon number 6 is the upper side of the carbon number 5, which you can see from this diagram. All right. When the examiner asks from you, you are required to draw it like this, not like that. Yeah, that the examiner can give you by in the question, but when they ask from you to draw like this, and mentioning that which number of carbon is that, like it's meant to carbon number 1, carbon number 2, 3, 4, 5, 6. Okay. All right. So where were we? In animals, glucose is the main transport sugar in the blood, and its concentration is in the blood is carefully controlled. Okay. There are many monosaccharides with the same chemical formula, C6H2O6. This is the same formula for all the monosaccharides, or for the all carbohydrates which having 6 carbons, because the general formula is the same. So wherever it's written, it means that this molecule is having 6 carbons. It contains 6 carbons, okay, but different structural formula. These include fructose, all right, like ribose, galactose, fructose, and glucose. These are having the 6 carbon. Means the general formula is the same. That how can you determine that whether which is fructose and glucose? So you're required to just focus on the diagram, which is the structure. Common 5 sugars, where n is equals to 5, include ribose and deoxyribose, found in nucleic acids, and ATP. ATP means adenosine triphosphate. Yeah, it means that ribose are 5 sugars. Ribose and deoxyribose consists of 5 carbons. Okay. Disaccharides are formed when two monosaccharides are joined together by a glycosidic bond. The reaction involves the formation of the water molecule. Okay. Remember one thing more. Okay. Wait a minute. All right, one more thing which was very important and I just forgot to talk to you guys is that, okay, now this is the structure of glucose. One most important thing is that you are required to focus one thing, which is alpha glucose and beta glucose. All right, there are two types of glucose. One is alpha glucose and other is the beta glucose. How can you determine from the structure while the formula is the same? And I taught you the fructose and glucose structures are different. In the glucose, how can you determine that the structure? By the structure? No, by the position of hydroxyl group. Remember this in your mind. All right. Okay, in front of you, which type of glucose is this? Alpha or beta? It's the alpha glucose. And how can you determine it? Due to the reason you are required to focus on carbon number 1, where the hydroxyl group is present. Below or above the carbon number 1. If it is present above the carbon number 1, then it is beta glucose. Alright, and when it is below the carbon number 1, then it is alpha glucose. Alright, plus what else is I want you from you guys to just remember is that in the diagram, as I have seen many of the teachers or many of the uh, Many of the YouTubers and many of the teachers who are just saying that you're required to just focus on carbon number one of where the hydroxyl group is present. If it is above the carbon number one, then it might be beta, and when it is below, it means that it is alpha glucose. The other thing which I want from you guys is to remember the whole structure where the carbon number two, where is the position of hydroxyl group at the carbon number two? Below. Carbon number three, above. Carbon number four, below. This is the pattern. And at carbon number 5, there is no hydroxyl group. Yeah, it is a hydroxyl group at the carbon number 6, you can say. Okay, and if you want to draw it, the carbon number 6 accordingly towards the branch. Okay, so you can draw it like that. We have just write simply, which is acceptable. All right. Now, why I am saying you as you, although it's very perfect, you are determining that carbon number 1. If the hydroxyl group is below carbon number 1, then it might be beta, it might be alpha, and if it is above the carbon number 1, it might be alpha or it might be beta. But why I'm saying you to remember the hydroxyl group positions at carbon number 2, 3, 4. This we'll be discussing in the later structures when we are just recovering with the structures of polysaccharides. All right. So where were we? Glycosidic bond. Okay. These both. Which type of glucose is these? Are these when they are combining? Okay. Which type of reaction is this? First of all, either is this hydrolytic reaction or a condensation reaction. Remember, whenever the water is in the product, it is the condensation reaction, all right, where the two molecules are joined together. Whenever the hydrolytic reaction is there, it means that hydrolytic reaction occurs. It means that the water molecule is combined with the reactant. Okay. Okay. So here now just focus on these both glucose. These are the glucose basically. Which one is beta and which one is alpha? Can you just tell me? By your thinking, just see or focus where is the hydroxyl group present, above or below. That's very simple formula. Okay, it is below the carbon number one. When it is below carbon number one, so what is this, beta or alpha? Beta? No, it is alpha. So these are both alpha molecules combined together. Understand? Now remember one thing that the candidate thinks, so that yeah, the both on the reactant, the both as you can see from the dotted line, the water molecule is removing from this molecule. You see, many candidates think so that no sir, it is alpha. Both are alpha due to the reason that carbon number 1 and carbon number 4 both have the hydroxyl group below. No, you're required to focus on carbon number 1. Alright. Where is the hydroxyl group? If it is above, it means it's beta. When it is below, it means it's alpha. Understand? So these are both the alpha glucose which are combined together to form a glycosidic bond and the water in the in the product. Here the glycosidic bond is present. Alright, the glycosidic bond form. This is called the glycosidic bond. There's carbon, oxygen and carbon bond. But not just this, as I was just thinking that yeah, that's enough to just memorize that glycosidic bond is carbon, oxygen, carbon, and the peptide bond is carbon, nitrogen, and this. No. We are required to focus on the whole diagram, whole structure. Okay, there is a glycosidic bond and the water is in the reactant. Okay, so this is how whenever the examiner will ask from you to make the diagram of condensation, glycosidic bond and the water in the product, you are required to draw it like that. Okay, I hope so that's easier. This shows glucose molecules joining together to form the disaccharide maltose. All right, it means that alpha glucose plus alpha glucose is equals to maltose. All right, because this bond between is between carbon number 1 of the one molecule and carbon number 4 of the other molecule, it is called a 1,4 glycosidic bond. You are required to just mention that on which carbon number 1, which carbon number the glycosidic bond is formed. Like here, number 4 are bonded together, which is the bond obviously, but you are required to write the which type of which number of carbon consists of this bond. Okay, plus the linkage. What is the linkage? We'll be discussing it later. Okay, and this kind of reaction where water is formed is called a condensation reaction. Yes, where the water is in the product. The reverse process when bonds are broken by the addition of water, such as in digestion, is called a hydrolysis or hydrolytic reaction. Okay, polymerization are condensation reactions. Yeah, we can say that polymerization or condensation reactions both means the same. And breakdown reactions are also hydrolysis reaction. There are three common disaccharides: lactose and sucrose. Maltose or malt sugar and is glucose and glucose. Alpha glucose plus alpha glucose. Remember? It is formed on digestion of starch by amylase because this enzyme breaks starch down into two glucose units. Brewing beer starts with malt, which is a maltose solution made from germinated barley. Maltose is the structure shown above. Alright, this is the structure of maltose, where the glycosidic bond is also shown to you. Alright, sucrose or canned sugar is glucose and fructose. Okay, it is common in plants because it is less reactive than glucose and it is their main transport sugar. It's the common table sugar that you put in the tea. Yeah, when you use the sugar in your home, these are the sucrose sugar, all right, or simple sucrose. Lactose or milk sugar is galactose and glucose, or we can say more easier that monomer plus monomer is equals to dimer. Galactose plus glucose is equals to lactose. It is found in mammalian milk and is the main source of the energy for infant mammals. Alright, discussing the polysaccharides. I hope so that, okay, this is the last thing, polysaccharides. Polysaccharides are long chains of many monosaccharides joined together by glycosidic bonds. Okay, poly means long or more, and saccharides, sugars. Starch is the, okay, one more thing which are required to remember is that polysaccharides are not sugars. Alright. Starch is the plant storage polysaccharide. It is insoluble and forms starch granules inside many plant cells. Being insoluble means starch does not change the water potential of cells, so does not cause the cells to take up water by osmosis. More on osmosis later. Okay, it is not a pure substance, but it is a mixture of amylose and amylopectin. We'll be discussing the starch structure later, and amylose and amylopectin are the both of the, what we can say, we are required to just understand these structures also. So this is the structure of amylose and amylose. Okay, in the amylose, where it's written that amylose is simply poly 1,4-glucose, so it's a straight chain. In fact, the chain is floppy as shown here, and it is tends to coil up into a helix. All right, it is the helical shape you can see here, where the 1,4-glucose are simply bonded. Okay, amylopectin is 1,4 glucose, okay, poly 1,4 glucose with about 4%, okay, 1,6 branches. Yeah, means you're required to have that there are 1,4 this is the 1,4 linkage including the 1,6 linkage which is at the branches. Okay, so what is the difference ones? 1,4 glucose simply bonded, and in amylopectin there are 1,4 and 1,6 linkages. Okay, this gives it a more open molecular structure than amylose because it has more ends. It can be broken more quickly than amylose by amylase enzyme. Okay, that's all. Both amylose and amylose are broken down by the enzyme amylase into maltose, though at different rates. Okay, and the glycogen, we'll be discussing glycogen. Glycogen is similar in structure to amylopectin. It is poly 1,4 glucose linkage with 9% 1,6 branches. Okay, it means that in one more different here is amylopectin has chain and branches, while amylose does not has branch. It only just have chain. Okay, glycogen is shown here. Okay, glycogen is similar in structure to amylopectin. It is poly 1,4 glucose with 9% 1,6 branches. Do not remember the percentage here. Okay, 9%, 4%. It is made by animals as their stored saccharide and is found mainly in muscles and liver. Because it is so highly branched, it can be mobilized. Mobilized veins can be moved or break down to glucose for energy very quickly. All right. One more said, what is glycogen? Glycogen is stored in liver and muscles. It is a stored form of glucose. You can say in the simple words. Okay, it is a stored form of glucose. Whenever the body requires energy, so the obviously there are the enzymes which break down the glycogen to glucose, which produces the energy. Okay, cellulose is one of the most important. Okay, cellulose is one of the most important here. It says cellulose is only found in plants where it is a component of cell walls. Okay. Do you know in your O levels, you have already discussed that cell walls are made up of cellulose? Yeah, cellulose is also a polysaccharide. Okay. It is poly 1,4 glucose 1,4 linkage, but with a different isomer of glucose. Okay. How it is different? Let's see how it is different. Glucose contains beta glucose in which the hydroxyl group of carbon on carbon 1 sticks up. What does it mean? Just let This means that the chain alternate glucose molecules are inverted. Okay. One for and one for glycosidic bonds in cellulose. What's the difference? Observe this difference here. I hopefully know that you may observe the difference here. The one difference which you can see from the 1,4 glycosidic bond in the starch, this is the structure of starch. You can see one thing here is that the glycosidic bond is below the carbon number 1 and carbon number 4. The glycosidic bond is below the carbon number 1 and 4. It means that hydroxyl group, obviously when the hydroxyl group will be removed and the hydroxyl group of carbon number 1 will completely remove, while hydroxyl group of carbon number 2 will carbon number 4 here only just the hydrogen will be removed and the oxygen will remain, which will be bond to carbon number 1 to make the glycosidic bond. You will be already observing that both glycosidic bonds are below the carbon number 1 and 4. The difference number 1 and carbon number 4, one glycosidic bond is above the carbon number 1 and 4 and one is below. It means that it is alternate. One glycosidic bond at up and then below, then above, then below, then above, then below, and it is repeated. Okay, this is the cellulose. All right, this is the structure of cellulose where the carbon's numbers are shown to you, not carbon written C is shown to you. Okay. Okay, now one more thing which I want from you candidates to focus is only just this example, this difference is visible to you candidates. I mean, okay, I'm giving you one more. What else is the difference here? There is a one more difference which is visible, may be visible to you candidates. Now just giving you the hint, focus on the numbers of the carbon. In the cellulose and in the starch, focus on the number of carbon. Do you focusing that the 6, carbon number 6 in the starch is always up above the carbon number 5. But in cellulose it is below carbon number 5. Although carbon number 5 is also below the ring. So can I say that they glucose molecules are inverted? Yeah, that's example. That's was a great hint. Even I have read the sentence completely. This means that in the chain alternate glucose molecules are inverted. All right. Why it is inverted? Okay, now this is the question. Why it is inverted? This is inverted to due to the reason that you are taking the beta glucose. But wait a minute, in the cellulose, all of the beta glucose are the same, obviously. And according to the information, we have already discussed that beta glucose means the carbon number 1 have the hydroxyl group above it. Okay? If the carbon number 1 hydroxyl group is above the carbon number 1, so then it means that this is beta glucose. So the question is that from beta glucose, every of the the glycosidic bond must be have to react to above the carbon. It's now above, then the glycosidic is below, then above. or why it is inverted. This is not visible here although, but I taught you guys that glycosidic bond is always formed between two hydroxyl group. As you can see from the example here, as in the starch, you that in carbon number 4 and carbon, when the beta is above, beta means above, the hydroxyl group is above the carbon number 1. It means that at carbon number 4, beta glucose must be below, right? Because of only just the reason that the beta glucose has the carbon number 1 above it, and on the carbon number 4 the hydroxyl group is below. So one is at above, both hydroxyl group of face each other to bond or to form the glycosidic bond. So that is why it is inverted, where the carbon number 4, which is having the hydroxyl group above it, will be reacting, okay, it's been reacting with the carbon number 1, all right. All right, so where were we? Yes, we were on the it must be essential to invert. Otherwise, can be not facing the hydroxyl group. The policy of glycosidic bond is that it will be only formed when the two hydroxyl group of each molecule which will facing each other at the same position. If one is at above, second must be on and above. All right, so that is why it is inverted, and that was a very easy question. You take a lot of time. Okay, this apparently tiny difference marks a huge difference in the structure and properties, obviously. While the 1,4 linkage glucose polymer in starch coils up to form granules, the beta 1,4 glucose polymer in cellulose forms straight chains. Hundreds of these chains are linked together by hydrogen bonds to form cellulose microfibrils. These microfibrils are very strong and rigid. Rigid means give strength to the plants, okay, and therefore to young plants. And this is the hydrogen bond shown to you. This is the cellulose, the structure of cellulose. Oh, that was my question from you, that which structure is that? But okay, no problem. I have to tell you guys that this is the structure of cellulose. How you identify, or how I identify, is that I saw that hydroxyl group, one is above, one is below, then above, then below, and this is repeated. And they are, because they are bonded to each other. The chains are bonded to each other through hydrogen bonded. And again, you can see the hydrogen bond is shown by the dotted line. Okay, the beta glycosidic bond specific cellulase enzyme. The only organism that possess a cellulase enzyme are bacteria. So herbivorous animals like cows and termites, whose diet is mainly cellulose, have mutualistic bacteria in their guts so that they can digest cellulose. Humans cannot digest cellulose, and it is referred to as fiber. Other polysaccharides that you come across includes the chitin, pectin, agar, murine, and lignin. Okay. Where they are found, you're required to just learn and just pause the video to see it. Okay. Okay, so we'll be discussing lipid in the next video. Until that, it's here. I hope so that you understand this video. Okay, so take care.